So I think we might start. Do you hear me all well? Okay. So good afternoon. We are very happy to welcome you to today's presentation, Are Dreams Meaningless? By Professor Mark Solms. My name is Helena Waldvogel, and I speak for the Student Initiative of Psychoanalysis at the Goethe University this evening.
And I am Terry Meyer from the Student Council, the Fachschaft of the Psychology Department.
Together, our groups have organized this talk by Professor Mark Solms, and financially we have been backed by the Freuds Beschreibung and the Astor of the Goethe University. So first of all, we would like to thank them for their support, and also we would like to thank Caroline Kula for designing the flyers and the posters for this evening. In view of Professor Tilman Habermas' imminent retirement, the question arises once again, which role psychoanalysis should play within university-taught psychology in the future. This question is of particular importance to us students, as it decides whether we will be taught psychoanalytic and psychodynamic theories by knowledgeable experts. With today's talk, we would like to spark and inspire discussions about how a fruitful interdisciplinary collaboration between psychoanalysis and more natural science-oriented subdisciplines like neuroscience can be created. We are convinced that both approaches, as different as they may seem at first glance, can benefit immensely from each other. And we think, after all, the hermeneutic theory of psychoanalysis offers insights that should not be underestimated by the university psychology with its mainly positivistic, scientific, and often quantitative research methods.
That is why we are particularly pleased to welcome the neuropsychoanalyst Mark Solms this evening here in Frankfurt, who in his research promisingly links psychoanalytic theory with neuroscientific methods. Mark Solms studied psychology and neuropsychology at the Witwatersrand University in South Africa. During his work on his doctoral dissertation, he trained to become a psychoanalyst at the Institute of Psychoanalysis in London. He has published more than 350 articles in neuroscientific as well as psychoanalytic papers, and is the author of 8 books. Currently, he's the director of the Neuropsychology Department at the University of Cape Town and supervises interns at the International Psychoanalytic University in Berlin. In his latest book, The Hidden Spring, he explores the question of consciousness and investigates the role of emotions. If I personally may add to that, last year, our students already had the pleasure to meet Mark Solms in a seminar that he gave on Zoom called Sigmund Freud Today. Indeed, a very fitting name for his research interests. In this course, Mark Solms really impressed and thrilled the students with his dedication to his work and his contagious passion for psychoanalysis. Hence, we are all the more pleased to have the opportunity to welcome Mark Solms here in Frankfurt in person today, and we're looking forward to his remarks today on the interpretation of dreams, and we are happy to give him the floor in a minute. Just one short remark regarding today's program. Professor Solms will be talking for about 60 minutes, and then there will be the possibility for a discussion of around 30 minutes. We will have the discussion. We can have questions in the audience. We can also have questions in the Canton audience as well as in the digital audience. And then I would ask you to type your questions in the chat later so that we can read them out to Professor Solms, preferably in English, but you can also try to translate them on the fly. And this meeting is being recorded, as you're probably realizing. And if you want a copy of the recording, you can just type your email address in the chat and then we can send it to you. Okay, so that should be that. Professor Solms, welcome, and please The stage is yours.
I believe that we're having some sound problems with the Zoom, and I apologize for that. But I'm very glad to be here at your beautiful university. Thank you for the invitation. I hope that in my lecture I will assist you in your struggle to have psychoanalysis represented at professorial level at this university. So I now begin. Subjective from them after they've woken up. So it's a retrospective report. And everyone knows how notoriously difficult it is to remember dreams. So it's a very unreliable second-hand retrospective report. But dreams, like the rest of subjective experience, dreams are a natural phenomenon. You all know from your own most direct empirical experience that they exist. They're part of nature. So we have to include them into science. The first person to seriously attempt to do so was Sigmund Freud. He developed the method of free association, whereby he invited his patients, and indeed not only his patients, he did it also on himself, to say everything that comes into their minds in connection with the images that they remember of their dreams. And Freud claimed to have found that by pursuing this method of subjective investigating the internal structure of subjective experience, he claimed to have found that if you follow these trains of thought, you find that they converge on a implicit theme, which is not manifest in the explicit imagery of the dream. And this implicit theme, claimed Freud, is always of the same kind. He said that dreams appear always to be driven by a wish, that they are an attempt to fulfill a wish. And what's more, the wishes concerned, says Freud, are of a rather base kind. They are perhaps better described as urges, something of the kind that we find driving the mental life of animals. This was a great shock to his contemporaries, the claim that we are driven by these base urges, the claim that these base urges motivate our dreams. And what is worse, says Freud, you don't see these things, you have to infer them through this method of following the subjective lines of associations of the dreamer. This is a very shaky scientific method. And it's therefore not surprising that not everyone was persuaded by the conclusions that Freud drew. That's why these things that emerge during dreams are not apparent to us during waking life. Freud said that there's this other system here between the unconscious memory systems and our consciousness. He called this the pre-conscious, and he said this is where we filter access to our innermost thoughts, wishes, and desires. And then it's from these more logical and rational and realistic thought processes, which are constrained by what is really possible and permissible in the real social world. It is from this that we then act on the basis of all of these things that we have learned, all of these perceptions which have left traces in the memory systems of our mind. So this is how Freud imagined the mind might be structured on the basis of what he observed of dreams, because he said it appears that in dreams, where motor action is blocked, it is not possible to act while you're sleeping. Your motor systems are paralyzed. He says it appears that this part of the mind, like your motor systems being blocked, this part of your mind goes to sleep. This is what sleep consists in, a loss of this reality-oriented part of the mind, what he later called das Ich, the I, me, the thing that I experience myself to be in control of my actions. He said that's what goes to sleep, but this deeper unconscious level does not go to sleep. It continues to press for discharge, for fulfillment. And he said because the path toward motor activity is not available, and because these motivational processes go backwards. They regress back onto the perceptual systems via the memory systems from which they were constituted, and there we have an hallucinatory experience, the dream. And Freud said the reason why we have this hallucinatory experience is if it were not for this discharge, this safety valve, as it were, where we have an imagined reaction, these heartfelt urges, desires, and wishes would press for motor action. And this is incompatible with sleep. And therefore, this re-representation of our wishes in this hallucinatory, imagined, virtual world of the dream protects sleep. So, said Freud, the function of dreams is to protect sleep. And he said, the reason why the wishes are not manifestly obvious, but rather are so disguised, is partly as a result of this regressive process, whereby thought processes get turned back into their raw materials. Abstract ideas get turned back into concrete perceptions. Not only this, but also there's a disguise process. They are censored and disguised, our dreams, because if we were to see what it is that we really want to do, this too would disturb our sleep. Dear students, I'm sure you can see this is a very big set of theoretical claims on the basis of really a very rough and ready research method. Said Freud at that time, firstly, what I just said to you at the beginning of my talk, dreams exist, subjective experience exists, it's part of nature. We need to adapt our methods to nature. We can't adapt nature to our methods. If there are subjective phenomena in the world, then we have to investigate them in their subjective form, using methods appropriate to the investigation of subjective experience. And Freud said, if anybody applies this method rigorously, they will find what I found. Behind every dream, if you follow the dreamer's thoughts, you will be surprised to find there is a heartfelt a wish of a basic kind. And he said, secondly, not only the first thing that these things exist and we therefore have to study them, psychological phenomena need to be studied by psychological methods. He said, but also one day in the future when other methods, physiological methods become available to study these things also neurologically, then we will join up our method with those methods. But there were no such methods available in 1900, which is when Freud published this theory. I want to repeat, because of the technical difficulties and interruptions, I want to make sure that I got across the main point, which is that using a subjective method of investigation, Freud came to the conclusion that dreams have a definite psychological structure. They're not just random noise. They are a form of mental activity occurring during sleep, which has a structure and a purpose. This purpose is to give expression to our wishes, which, if they were to be acted upon in reality, would be incompatible with sleep. Therefore, they regress onto our perceptual systems in an hallucinatory form. And there we imagine ourselves to be fulfilling our wishes, thereby protecting sleep. And they are disguised to further protect sleep, because if we were to confront our innermost desires, it would cause us anxiety. So that's Freud's claim on the basis of this method, and that's where things stood for a very long time. Freud's ideas about dreams were very influential. He built the whole of psychoanalytic theory upon his theory of dreams. This drawing on the screen, as I said earlier, was his first attempt to infer what the structure of the mind as a whole might look like, based on what he had learned about the apparent structure of the processes giving rise to dreams. Psychoanalysis, starting in the German-speaking world, in Vienna and the Austro-Hungarian Empire and in Germany, it spread, especially after the Second World War, all over the world and became extremely influential in psychiatry. For example, in American psychiatry, after the war, it was really quite impossible to obtain a chair of psychiatry at a decent American university if you were not a psychoanalyst. You were considered to be not properly qualified to practice psychiatry and teach it if you were not a psychoanalyst, because psychoanalysis was the basic theory that was applied in all of psychiatry. Remember, this was an embarrassing situation because all of it was based upon this method, this very shaky method, which was all that we had, the only way that we could go about investigating something so subjective as dreams. But philosophers of science were not so sure that these conclusions that Freud drew using the method that I've described, that they could be considered valid scientific conclusions. And this view was expressed most forcefully and most influentially by another great Viennese mind, Karl Popper. Karl Popper, from the 1930s onwards, started to articulate the view, and ultimately it culminated in the book, the cover of which I have on the screen, Conjectures and Refutations. Karl Popper came to the view that a scientific theory, to be considered scientific, must give rise to falsifiable predictions. In other words, you must say, if such and such, if I do such and such in an experimental situation, then on my theory, the following outcome will occur. In a situation where it's possible for another outcome to occur, which shows your theory was not correct. What you predicted would occur is contingent upon your theory, and if that does not occur in this critical test, then your theory has to be revised. As long as your prediction is confirmed, then your theory is held to be provisionally true until there's further experimental evidence where predictions are not confirmed and which lead us to revise the theory. This is the basic philosophy of science that just about all natural scientists follow this philosophy. They agree with Popper. For a theory to be considered scientific, it must give rise to falsifiable predictions. Now, in this very book that's on the screen, that was so enormously influential in the philosophy of science and indeed in science itself, Karl Popper uses Freudian theory as an example of what is not a scientific theory. It looks like a scientific theory, it sounds like a scientific theory, says Popper, but it is not, because it does not give rise to falsifiable predictions. Popper made the point that the theory claims, first of all, that dreams are giving expression, or at least they're an attempt to express repressed wishes, and secondly, that this is in the service of protecting sleep. But the evidence for this is remembered imagery from the dreams, which do not reveal the repressed wishes. The only way to gain access to the repressed wishes is this method of reassociation, which Freud said leads you to observe that there is an implicit psychological structure behind those associations, which takes the form of a wish. And how do you find the wish, therefore? You have to infer it. In fact, the method that Freud used was called interpretation, Traumdeutung. And, said Popper, this method of Deutung how do you, if the analyst says, well, behind there, I infer, I can discern behind the material a repressed wish. How can you ever falsify that? Says Popper, the analyst can always find a repressed wish because this method of interpretation allows it. You can't say, well, I don't see a repressed wish. The analyst says, well, that's just because you're a bad interpreter. Yeah? And so, said Popper, he used this word, I'm afraid to say, he said, so the psychoanalytic theory is pseudoscience. It looks like science, but it ain't necessarily so. By the way, not many people seem to know that Popper also said, actually, I believe that Freud is right, but nevertheless, the theory is unfalsifiable, and therefore we cannot consider it to be a proper scientific theory. So you can imagine the consternation in American psychiatry when the great Karl Popper says it's based on a pseudoscience. And remember that the interpretation of dreams was foundational to the whole of psychoanalytic theory building. It therefore, no doubt, came as an enormous relief when, in the early 1950s, two physiologists in America, in Chicago, Azarinsky and Kleitman, applied an objective physiological method to the study of what's going on in the brain during sleep. They predicted, this is an EEG, it's a little cartoon of what an EEG looks like. On the x-axis, we have time, 8 hours of time. And on this axis here, we have depth of sleep as measured by the level of brain arousal on the EEG. The EEG has short little fast waves when the brain is very aroused, and then they become slower and slower as you go more deeply into sleep. And so, as Azarinsky and Kleitman predicted, that as you fall asleep, so your brain waves will slow down, and that's exactly what happened. Their prediction was confirmed. It was a reasonable prediction, not a great surprise that the brain arousal decreases as you fall asleep. That's what falling asleep seems to be all about. The great surprise was that what happens next is the brain then becomes aroused again, almost to waking levels. And then it goes back into deep sleep. And then it becomes aroused again, and then back into deep sleep. Azarinsky and Kleitman called this arousal of the brain during sleep paradoxical sleep. The paradox being, how come your brain is aroused when you're asleep? This led them to the, again, not unreasonable hypothesis that perhaps this is when the brain is dreaming. The arousal of the brain is it's booting itself up again in the generating of dreams. So they tested this hypothesis by making the falsifiable prediction that if you wake people up from this paradoxical sleep, they should report dreams. And if you wake people up from this deep sleep, also known as slow wave sleep, then they should not report dreams. And that's exactly what they found. Their student, William Dement, the unfortunate name of Dement, he did the study and found if you wake people up during this phase of sleep, which later became known as REM sleep after rapid eye movement, because it's not only that the brain is aroused in this state, it's also that your eyes dart about and your muscles below the neck are completely paralyzed. That constellation we call REM sleep. If you wake people up from REM sleep, you get dream reports roughly 90% of the time. And if you wake them up from non-REM sleep, from deep sleep, from slow wave sleep, you get dream reports 10% of the time, if not less. So that's about as close as you could hope to come to a confirmation of that prediction, given all the vagaries of sleep that I told you, of dreams that I told you about earlier. To have a 90 to 10 confirmation that REM sleep, you have dreams 90% of the time, non-REM sleep, you have dreams 10% of the time, was as good as you could expect to get. And so from that moment onwards, which was 1955, when Dement's paper was published, it was generally accepted that dreaming is the subjective experience of REM sleep, an objective physiological state. And why this was such happy news was because at last, in the study of dreams, which was so foundational to the whole of psychiatric theory, based as it was those days on psychoanalytic theory, we now at last could apply objective methods. We can wake people up when we know they're dreaming. We can get direct access to the dreams. But much more importantly, we now have a physiological objective biomarker which doesn't just identify when we are dreaming, but enables us to discern what the brain mechanisms are that generate this subjective state. In other words, what is going on objectively when you're having these subjective experiences. I told you subjective experiences are an embarrassment to science. So now we could drop them and study the objective side of the equation. This was made all the more viable by virtue of the fact that it's not only human beings who display REM sleep. All mammals display REM sleep. On the screen here, I have a picture of a human brain, but the research program that was triggered by Dement's findings in 1955 was an attempt to find out which part of the brain generates REM sleep by systematically damaging the brain and seeing which damage where leads to a loss of REM sleep. That will enable us to discover what causes REM sleep and therefore dreams. So although I have a human brain on the screen, the research was done on cats. Why they chose cats, I can't imagine, apart from the fact that their brains are very similar to ours, so that the brain on the screen looks very similar to a cat brain, and perhaps also because cats sleep so much. So these poor cats, Michel Jouvet started systematically to damage the brain, going deeper and deeper down, until eventually he cut it here. He severed the entire forebrain, he severed it, disconnecting it from the brain stem. And in these cats, REM sleep continued unabated. What that showed was that the forebrain, the organ of the mind, the part of the brain which contains those memory systems, those perceptual systems, those desires that Freud spoke of, all of these psychodynamics. Imagine the complexities of the mental processes involved in disguising and censoring your dream processes so as to not disturb yourself, etc. All of that, said Jouvet, is not involved in the causal mechanisms of generating REM sleep and therefore dreams, because you can disconnect the organ of the mind, the forebrain, entirely, and yet REM sleep continues in the same way. So he cut deeper and deeper, and it was only once he got down here to the pons, the Brucker, once he got down there, which is really a very low part of the brain. Beneath the pons is the medulla oblongata and then the spinal cord. This is a part of the brain that we share with all other vertebrates. It looks the same in a fish, just a little bit smaller than yours, but it's got the same basic design. So it was a real shock to discover that dreams are generated because REM sleep is generated at this very lowly level of the brain. Jouvet published his findings in 1965. He then passed on the baton to his student, Alan Hobson, who in much more detail discovered exactly what's going on within this part of the brainstem, the pons. And he found that there's a group of cells over here called the mesopontine tegmentum, which start firing rapidly as you enter REM sleep. They fire normally, tok, 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 and then they go tok, 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 as you enter REM. And then the other cells down here in the locus ceruleus complex and the dorsal raphe nucleus, where the opposite happens. They're normally firing at a high rate, and then as you enter REM sleep, they go. So what switches REM on is these cells, and what switches REM off is these cells, said Hobson. These cells are the source cells of a neurotransmitter called acetylcholine. Acetylcholine, from the cell bodies here in the mesopontine tegmentum, they send long axons up to the forebrain, and there they arouse the entire forebrain. So, says Hobson, when you switch these ones on, you switch on dreams. At the same time, you switch these ones off. And conversely, when you switch these ones on, you switch these ones off, and that switches dreaming off, REM sleep and dreaming. So what is the mechanism whereby dreams are caused? Said Hobson in a famous paper published in 1975. It's a simple switch, on, off. And what is more, that switch is switched on and off in a part of the brainstem where there's no psychological nothing going on. To quote Hobson, he said, it's motivationally neutral. What is more, please remember, this switches on with regularity. You saw those cycles during sleep. Every 90 minutes, like clockwork, you go back into REM sleep, back into REM sleep. So it seems to be a pre-programmed automatic physiological state. And Hobson wasted no time in drawing the conclusion that therefore dreams cannot possibly work the way that Freud claimed they do. 1 year later, he delivered his findings to the American Psychiatric Association, which had previously been so dominated by psychoanalysts. And there he presented a theory of how dreams work, which was called the activation synthesis theory. He said, these cells in the mindless, motivationally neutral brainstem automatically, in a pre-programmed regular clockwork fashion, switch on. Then they randomly activate the forebrain, which is where your memories and perceptions and desires and intentions and beliefs and so on are located. And to quote Hobson, the forebrain makes the best of a bad job. It joins all the dots and it turns it into an experience. Said Hobson, the reason why that experience is so bizarre, so unrealistic, so unlike your normal waking cognition, is because it is generated by random noise. The brainstem activates the forebrain diffusely. Normally, the forebrain is activated by patterns of stimuli arising from the outside world, representing actual things that are happening there, interacting with your memory systems of previous such things that have happened to you. So that's meaningful. But this is chaotic, random noise coming up from the brainstem. So it's meaningless. And the forebrain, to quote Hobson, makes the best of a bad job by joining up those dots. Hobson made the telling point that you can, of course, afterwards go and look at that random noise, this froth, this nonsense that you dream, and you can project meaning into it in the same way as you can project meaning into an ink blot. You can say, ooh, this ink blot makes me think of bats or butterflies. And if it makes you think of bats versus butterflies, it might say something about you. But the important thing is that the meaning is not in the ink blot. You have projected the meaning into an utterly meaningless ink blot. It's intrinsically meaningless. And Hobson said, on the basis of his findings, dreams are intrinsically meaningless. We retrospectively project meaning into them. And when he presented these findings to the American Psychiatric Association, a vote was taken by the assembled membership as to whether Freudian dream theory was still viable or not. And the conclusion reached was by a margin of 2-1 that Freudian dream theory was no longer scientifically viable in light of these findings. That was a momentous event in the history of psychoanalysis and its status as a science in psychiatry and the mental sciences in general. That paper was published 1 year later in 1977. This was 3 years before I came into the field. And when I studied neuroscience, neuropsychology, that branch of neuroscience which most interested me, I was interested in this idea that the brainstem activates the forebrain, and then the forebrain synthesizes all of this activity, synthesizes it into a dream. That was the activation synthesis theory. By the 1980s, we knew quite a bit about what the forebrain does. And the idea that what it does is synthesize was a little bit, I thought, too generic a description of what cognition consists in. I mean, we know, for example, that the frontal lobes have an overarching executive control of cognitive processes, that those cognitive processes include visual material arising from this part of the cortex, auditory material from here, somatosensory from here, motor imagery from there, language on the left, et cetera, et cetera. So my interest in this theory of Hobson's was to try to get a little bit more flesh on those bones, to try to understand better what the forebrain's contribution to dreams is. I predicted, for example, that this part of the brain, the prefrontal lobes, which are normally in charge, it's normally your volitional a voluntary, reflective, logical, realistic self that's normally in charge of your thought processes, it seems that this part of the mind is not very active during dreams. You don't seem to be in charge of what's happening. It all just happens to you, and you can't influence things and so on. And they're not rational and reasonable and goal-directed and so on. So I said, well, perhaps damage here will not have much effect on the content of dreams. As opposed to damage here in the visual parts, dreams are dominated by visual phenomena. So I thought, well, damage here might lead to a total loss of dreaming, or at least to a loss of the visual quality of dreaming. I wondered whether damage here in Broca's area might lead to a loss of the ability to speak in dreams, as it does in waking life. So I was wanting to get a more variegated pattern. So I predicted these things. Damage here will lead to nonverbal dreams. Damage here will lead to paralysis in dreams. Damage here will have no effect on dreams. Damage here will lead to non-visual dreams, or perhaps to no dreams at all. I wasn't studying cats, of course, because I had to study the subjective experience of the dreaming brain. So I had to study human beings. Needless to say, I therefore couldn't systematically damage their brains to see what would happen. I'm not so sure we should ever systematically damage any animal's brain to see what will happen. But you certainly couldn't do it, and still can't do it, on human beings. So I had to wait for naturally occurring damage, tumors, strokes, and the like. And so it took me several years to collect my data. I started in 1985, and I published my first findings 10 years later in 1995. I collected a sample of 365 patients with focal damage, groups of patients with focal damage to different parts of the brain. Here, this slide summarizes my findings. I'm not going to tell you about all my findings. It'll take us all night, and we're running late now as it is. But I'll tell you, the one prediction that I made that was confirmed was that damage in this dark blue area, the prefrontal lobes, damage to the prefrontal cortex, to this part of the brain here, does have no effect on dreams. Although these patients are severely impaired in their waking cognition, blind raters cannot tell the difference between their dreams and yours and mine. They're the same. That was the only prediction I made that was confirmed. The other findings were all surprises. So let me go through the surprising findings. The first was that damage here in the mesopontine tegmentum, damage here in the part of the brain that generates REM sleep, although in us humans, just as in cats and rats and all other mammals, damage there leads to a loss of REM sleep. Every single one of my patients with damage there who were conscious, because many of them lose consciousness, they go into coma. But those who were conscious, I had 18 of them. All of them reported preservation of dreams. If you wake them up in the sleep lab, you can't wake them up during REM sleep because they don't have any REM sleep. But if you wake them at random intervals, you get no dream reports at all, consistent with their morning report that they don't dream, that they dreamed nothing last night. Now, this was a really big surprise because it is one of the ABC rules of behavioral neuroscience that if you're going to say a particular function, in this case dreams, is generated in a particular part of the brain, in this case the mesopontine tegmentum, then loss of the mesopontine tegmentum should lead to loss of dreams. But it didn't. It led to loss of REM sleep with preservation of dreams. This suggested, was the first hint that maybe dreams and REM sleep are not the same thing. I then, because it was such an astonishing finding, I went back to the literature because remember, REM sleep had been discovered 35 years before I started my research. And that entire 35 years of literature, I could not find one single case in which loss of dreaming had been reported as a result of damage to this part of the brain. There were a good few cases of human beings in which there was loss of REM sleep reported in the literature, but none of them reported loss of dreams. And in fact, the majority of them, the investigator did not even ask. You must remember, subjective phenomena are an embarrassment to science. We were interested to be able to objectively demonstrate that in human beings, just like rats and cats, damage there leads to loss of REM sleep. Either nobody thought to ask them, or maybe they did ask them and it would weaken their conclusion if they to say, look, we're the same as these other animals, So whatever the reason may be, it was never has a damage there been reported to lead to loss of dreaming, just like in my case. So I thought, well, gosh, this is quite something. The second finding, which was a big surprise, is marked in the brown areas here. I found that patients with damage to the parieto-occipital region over here, that damage there led to a complete loss of dreaming. In these cases, REM sleep was preserved. So this is what we call a double dissociation of function. With damage to one area, you have loss of REM sleep with preservation of dreams. With damage to another area, you have loss of dreams with preservation of REM sleep. That is a double dissociation, which indicates that these are two separate functions. They are dissociable and they are subserved by different brain mechanisms. That is really quite astonishing. It is a very major error that we made. And I want to pause for a moment to contemplate with you what the basis of our error was. How could it be that on the basis of observing this correlation of dreams and REM sleep in the mid-1950s, we then studied only the REM sleep side of things and its brain mechanisms and didn't investigate the subjective side of things? Well, the main reason why was because we focused our neuroscientific studies on other species for the methodological reasons that I've already described. But I think it's also because we don't want to study the subjective side of things for the reasons I've already described. Once you've identified the objective correlate, this correlation has been established in dementia studies, then we wanted to study only the objective mechanisms. And we wanted to drop like slippery fish these subjective, retrospective, second-hand reports from people who've been asleep when they had the original experiences. And it led us to this. I want to be clear about what the mistake was. I'm sure you all learned it in your first year of psychology, that you must not confuse correlation with cause. Because these two things happen together, does not mean that the one causes the other, let alone that they are, in fact, the identical thing, which is what the claim was. Hobson explicitly made the claim that the subjective experience of dreams is the same thing as the objective mechanism of REM sleep. We're studying the same thing from an objective versus a subjective point of view. You see it manifests in these two different ways. That's to claim that because there's a correlation between dreams and REM sleep, they're therefore the same thing, and to continue in the way that they did with REM sleep research, is the same as to say babies cry when they're hungry. Therefore, if you want to understand the mechanism of crying, we must investigate the digestive tract. It was exactly an error of that kind. So with my study, all that I had added was just to actually ask my patients, are you still dreaming? Well, in fact, I said to them, how do you sleep? And then I said to them, once they'd answered that, and what about dreams? And then unless they said to me themselves something that they no longer dream, I said, do you still dream? And if they said they no longer dream, I said, did you used to dream? And if they said they did, I said, when did you stop dreaming? And if they said it was when I had my stroke, then I included them in my study and put them in the sleep lab to investigate what's going on with REM sleep and its relation to this apparent absence of dreaming. And all that I added, I have to emphasize, was to talk to my patients about their subjective experience. By leaving the subjective experience out, we made this really quite embarrassing error of confusing correlation with identity or causation. Now, I want to tell you about one further finding, which became the main focus of my research after that. It's the area I've marked in red. Red means important. This part of the brain is the white matter underneath the cortex here. There are fiber pathways that course through the brain over here, and damage to those fiber pathways, here I'll show you the scans in my cases, damage to those fiber pathways leads to a loss of dreams. So just as I found with the parieto-occipital cases, the cases with damage in the brown areas there, the patients with damage here, that they had preservation of REM sleep with loss of dreams, so too I found with these patients who have damage to the white matter deep inside the ventromedial quadrant of the frontal lobes. Why this was of so much interest was it wasn't so hard to understand why damage here to this part of the brain, the parieto-occipital region, is indeed where our mental imagery is synthesized. It's where visual and auditory and somatosensory perceptual inputs overlap, and it's what generates our heteromodal, multimodal mental imagery. It's not a surprise that if you have damage to the part of the brain that generates mental imagery, that you will have difficulty generating a dream. But it's by no means clear what this red part, these fibers coursing through the bottom part of the frontal lobe, why they should be so essential for dreaming. That's why my focus moved to this area. Now, remember what we're doing now is we're formulating hypotheses so that can lead to testable predictions. So my hypothesis as to what was going on here was based on the fact that when I looked at these scans of my non-dreaming patients with damage in this second area, I remembered that this is exactly the area that used to be targeted in the very old bad days by psychosurgeons. They performed an operation of cutting those fibers in psychotic patients. The operation was called prefrontal leucotomy. And the purpose of doing this operation was for the treatment of positive psychotic symptoms, hallucinations and delusions. As horrifying as it might seem, this was a successful operation from the point of view of controlling hallucinations and delusions. And that's why it was performed on hundreds of patients. In fact, I went back to the old literature to see whether in those old clinical series, in the 1930s and 40s and 50s, when psychiatrists were still interested in dreams, and psychiatrists were the ones who were sending these patients for these brain operations, they studied the effects of the operation. And I thought they might have asked their patients about their dreams after the operation. And I found in large series of cases, one by Jerome Frank of 500 cases in America, one by Partridge of 350 cases in Great Britain, and one by Schindler here in Germany. Again, a good few hundred cases. They all reported the following effects of this operation. Firstly, it leads to loss of hallucinations and delusions. Secondly, it leads to an amotivational state, a loss of spontaneity and initiative and drive. And thirdly, it leads to loss of dreams. So I hadn't discovered anything new. In fact, it had been reported decades before in hundreds and hundreds of cases that severing those fibers leads to a loss of dreams. In fact, Schindler made the interesting observation that if the patient after the operation continues to report dreams, it's a bad prognostic sign. He said it means the operation hasn't worked. And on reflection, you can see why that might make sense. Because if you can generate a dream, then you can generate an hallucination and a delusion, because that's what a dream is. Because one of the reasons why Freud was interested in dreams is because they were a psychopathology of everyday life, a state of insanity, of psychosis that we enter every night. So it was on the basis of that observation of Schindler's that my interest turned to this pathway. It's called the mesocortical mesolimbic dopamine system. There are many pathways that course through this part of the brain where damage leads to loss of dreaming. But I focused on this one because the operation that used to be performed, prefrontal leucotomy, was abandoned not for ethical reasons, but rather because a drug was developed which blocked dopamine activity in this circuit. Antipsychotic drugs in the 1950s blocked chemically activity in this circuit, which was previously severed surgically for the treatment of psychosis. It's clearly a preferable treatment over the operation. And so antipsychotics replaced prefrontal leucotomy. It's because antipsychotics acted on this pathway that I thought perhaps this is the one that generates dreams. So remember, that's a hypothesis. Now you have to test it. You have to test it with generating falsifiable predictions. I predicted that if you block dopamine chemically, you should reduce dreams. If you should find fewer dreams, shorter dreams, less emotionally intense dreams, less bizarre dreams, less nightmares. And conversely, if you boost dopamine chemically with a drug, you should find more dreams, longer dreams, more bizarre dreams, more emotionally intense dreams, and more nightmares. That's what we predicted, and that's exactly what we found. And interestingly, the boosting of dopamine, which leads to the boosting of every dream variable that we measured, does not lead to any increase in the REM sleep commensurate physiological correlate. Which, remember, we still have to explain why does dreaming happen at the same time as REM sleep. But clearly, we were boosting something, dopamine, which boosts dreams, but does not boost REM sleep. REM sleep, let us not forget, is generated not by dopamine, but rather by acetylcholine. And so it's interesting to observe that if you block acetylcholine, if you block dopamine, you block dreams. If you block acetylcholine, the rival hypothesis, the prediction should be that should lead to a loss of dreaming or a reduction of dreaming. It does lead to a reduction in REM sleep, but it does not lead to a reduction in dreaming. In fact, these patients paradoxically have increased dreaming, and some of them even dream while they're awake. In other words, they start to have hallucinatory experiences during waking life. Anticholinergic drugs do that. So on the basis of that study, that was the first confirmation, in addition to the lesion evidence, that this might indeed be the pathway that generates dreams. Another method led to the same conclusion. This was not a study performed by me, but rather by my colleague Alan Brown in Washington. He did PET imaging of the brain. When you go to sleep, what falls asleep is your frontal lobes more than anything else, the prefrontal lobes. And then when you go into dreaming sleep, the prefrontal lobes stay switched off. But what switches on, like a Christmas tree, is this circuit, the one that I spoke about earlier. In fact, all of the emotional and motivational circuitry of the brain, known as the limbic circuitry, all of it switches on very intensively during dreaming sleep, but none more intensively than the mesocortical mesolimbic dopamine system. There you see it, there you see it, there you see it. Interestingly, what also happens is this activation of the perceptual systems. So the executive frontal lobes are switched off, motor activity is switched off. There's this massive activation of the motivational, emotional systems of the brain and the memory systems of the hippocampus here. And there's a backward activation of the perceptual cortex, giving rise to the hallucinatory experience. So that's three methods suggesting that the part of the brain that generates REM sleep is here, that generates dreaming, and not REM sleep, dreaming is here. A fourth method was a study conducted by this colleague of mine. He did single cell recordings of those dopamine neurons and showed that they fire at their maximal rate during dreaming sleep. They fire in bursts, which leads to the maximal release of dopamine. And this colleague, she showed by another method, microdialysis, that indeed dopamine is released maximally. She measured the release of dopamine in dreaming sleep. So there's maximal release of dopamine at the terminals of that circuit during dreaming sleep. The single cell recordings show maximum firing rates of those cells during dreaming sleep. Brain imaging shows massive activation of that circuit during dreaming sleep. And drug trials show that you can upregulate and downregulate dreams by upregulating and downregulating the chemistry of that circuit. So on the basis of these multiple methods, multiple lines of converging evidence led us to the conclusion that the part of the brain that generates dreams is that circuit. So now the question becomes, what does that circuit do? Because clearly it is fundamental to what dreams are all about. Jaap Panksepp, who is one of the great experts on that system, he calls it the seeking system. His colleague Berridge called it the wanting system. Edmund Rolls and many others call it the brain reward system. What they all agree is that this is the single most powerful motivational desire and reward system of the mammal brain. As Panksepp says here, it generates the most intense, most energized exploratory and search behaviors that an animal is capable of exhibiting. And Dahan, who did the single cell recordings, said that it looks from the recording records the same as when the animal is getting its most intense rewards, its favorite foods, for example. So all of these different words for this system show seeking, wanting, reward, show that this is an all-purpose, pleasure-seeking. In fact, if there was any one part of the brain that you say, that you would ask any affective neuroscientist, anybody who knows about the brain mechanisms of affect and motivation and emotion, say which part of the brain is most like what Freud described as this drive, this motivational urge that drives dreams. They would say, well, the mesocortical, mesolimbic dopamine system. That is the most similar to Freud's libidinal drive of all of the brain's motivational and emotional circuitry. So I published my findings, as I said, in 95 and then in 97, and I was invited to speak at Alan Hobson's lab at Harvard. He wrote a very favorable review of a book I had just published, even though it was a real surprise to him to find that the part of the brain that generates dreams is not the part that generates REM sleep, that it's far from motivationally neutral. This dopamine circuit is the big-ticket motivational system of the human and mammal brain. Nevertheless, he wrote a favorable review of the book and invited me to speak to his team at his lab. I presented my findings and was cordially received. And at the end, I said, half jokingly to Alan Hobson, perhaps you owe Freud an apology. And he said, why you say that? I said, well, what I just said to you. I said, well, Alan, if there's any part of the brain that looks like Freud's drive that motivates the dream, it's there. And so, the reason that you gave for why Freud must be wrong was itself wrong. Dreams are not driven by REM sleep. And the part of the brain that does generate dreams looks pretty much like what you would have expected from Freud's psychological studies. I got an email after that visit, which I have printed out and kept for posterity, in which Alan Hobson said, did I hear you right at the end of the talk? You said that your findings, you interpreting them as supporting Freud. I said, because if that's how you're going to interpret your findings, then I am not going to support what you found. Oh gosh, this is a negotiation about, we all know what the findings are. But if you're going to say they support Freud, then I'm going to say, I don't recognize your findings. So much for objectivity in science. So that was a shocker. Alan Hobson and I were then invited to debate our respective views at the next Science of Consciousness conference. They have one every 2 years in Tucson, Arizona. I think this was in 2006. And I thought we were going to debate our respective views, but Hobson said he'll only take part in the debate if I defend Freud's views. In other words, I must defend a theory that by then was more than 100 years old, rather than defend my own theory, which was just a few minutes old. You can see it casts me in a bad light to be the one who has to defend the now discredited Freud against the modern scientist. So I agreed to take part in the debate on those terms, but started my own presentation by saying, look, I said to Alan, I think my findings are compatible with Freud's theories, but I'm not here to defend Freud so much as just to defend my own views, and then to say to you, I think that we owe Freud an apology on the basis of the fact, everything I said to you earlier. So at that meeting, 30 years after the one at the American Psychiatric Association, again, there was a vote taken, and this time the vote went 2-1 in the opposite direction. Our assembled neuroscientific colleagues concluded that Freudian dream theory was scientifically plausible in light of these findings. However, I really need to emphasize that this is just a matter of plausibility. It looks consistent with what Freud had inferred from the study of the subjective experience of dreams and the subjective associative trains of thought that flow from the different elements of the dream. It is really quite astonishing that by that method, Freud was able to come to such extremely unlikely conclusion that if you follow the thoughts of the patient, what you find is behind these dreams, implicitly, there is a wishful, heartfelt, wishing, wanting, desire process. It's really quite astonishing that this evidence here, which doesn't require any interpretation, I mean, there you have it on the screen. This is not implicit, it's absolutely explicitly visible that the dopamine circuitry of the brain is switched on full blast during dreaming sleep. It is remarkable that the psychological investigations led Freud to what seems to be the right sort of ballpark. But it is not a proof of the Freudian theory. It's just saying, well, what we thought was incompatible with Freud was itself wrong. This is compatible with Freud, but that doesn't by any means prove that Freud was right. Therefore, to come to the end of my talk, I want to tell you about a study that we are now busy doing. In fact, we've done the pilot phase of the study, and I'm going to show you the data from the pilot phase. And we are now conducting a much larger study at the Charité Hospital in Berlin. In fact, it's a colleague from your great city, Tamara Fischmann, who is conducting this study with me at the Charité. This study is designed to test a falsifiable prediction from the Freudian dream theory. Now that we know what the brain mechanisms are, now that we have this picture of the objective correlates of the dream process as opposed to the REM process, it enables us to test specific parts of the theory. Let me remind you, when I said it's remarkable how close it fits with the old Freudian theory. It seems like the executive part of the brain goes to sleep. The motivational part of the brain is switched on like a Christmas tree, and it regresses onto the perceptual systems. And Freud said this is in order to protect sleep. So what we see in the dreaming brain is that the prefrontal cortex switches off there, that switches off as you go to sleep, and it stays switched off as you enter the dreaming state. This deep motivational urges part of the brain switches on, and then there's a regression and an activation of the perceptual systems, generating an hallucination, which becomes impossible if that part of the brain is damaged. Remember the parieto-occipital region damaged there with loss of mental imagery. They also have loss of dreams. So bearing in mind that this is the most intense motivational circuitry of the mammal brain, it is really quite remarkable that we do stay asleep while we are so motivated. As Panksepp said, the most intense exploratory behaviors an animal is capable of. Daran saying it's the most intense reward pattern of activation. Like when the animal is getting its most intense rewards during waking life. With all of this motivational surge happening during sleep, how do we stay asleep? Freud said we stay asleep by having the dream that regresses onto the perceptual systems. Rather than real action, we have this imaginary action. It's a very simple theory. It's a very simple claim, now easily testable. So the prediction is patients who have damage to this parieto-occipital region of the brain and who therefore can no longer dream, when they have this motivational surge in the middle of the night, rather than their sleep being protected by a dream, it should wake them up. It's an obvious falsifiable prediction arising from the theory. Do you get my reasoning? So that was the study that we did, the pilot study that we've completed. We took patients who had had a stroke to the same arterial territory at the back of their brains. In all of them, it was a thrombotic infarction, which makes for a very neat area of damage to the brain. And we compared those of them who had damage there who did not dream with those of them who had damage there who did dream. Now, obviously, there's a slight difference in the anatomical location of the lesions. There are a few centimeters in question here. And we found that the lesions are slightly more anterior, overlapping with, as in those who don't dream, than the lesion slightly more posterior in those who do dream. But there's a strong overlap between the two. Everything else was identical. They had strokes of the same kind. They were matched for age. They were matched for the duration since the stroke. They were matched for absence of paralyses, etc., etc. Everything was controlled. The only difference was the main experimental variable, do they dream or not? Tested in the way that I've described to you before by waking them in the sleep lab and so on. So now I'm going to quickly, because we're running late, I'm going to quickly give you the results of this study. before I give you the results, I want to make sure that you understand what matters about them. What matters is not what we find. So if we find that the patients who cannot dream do not have worse sleep than the patients who can dream, then we have falsified the Freudian theory that dreams protect sleep. What's important is that we can falsify it. It is a theory which, using the appropriate methods available to us today, not available in Freud's time, exactly as Freud predicted, one day, we've learned a great deal by studying the subjective phenomena of dreams subjectively. It's not that that's worthless. It made us understand something of what the psychological processes that lie behind dreams are. But in the future, it will be possible, we assume, to link up with neuroscience and study these things objectively. So, and remember, you can't study them objectively alone. Dreams are subjective phenomena. Remember the mistake we made by leaving the subjective side out. Why can't we use both? Every method has its strengths and its weaknesses. The psychoanalytic method, studying the rich complexities and dynamics of experience, has great strengths. It also has terrible weaknesses, as Popper pointed out. But the neuroscientific methods have their great strengths in terms of the objective and precise measurable rigor. But look at the mistake we made by leaving the subjective data out. So, we want to include both. And so I say again, for me, the important thing is that using the appropriate combination of methods, we are able now to test this theory, test predictions arising from this theory, and falsify them if they are wrong, if the theory is wrong. So remember, what I'm going to show you now is what we found. That's less important than the fact that Freudian theory is not pseudoscience. The theory itself gives rise to falsifiable predictions if you use the appropriate methods. The theory is derived from one method and can be falsified by another method. That's the essence of what I'm wanting to show you tonight. So let me say again, our prediction was that patients with damage here, because their motivational urge and surge still carries on during sleep, because they have damage here, if they can't dream due to that damage, they should have a poor quality of sleep, which is the opposite of what you would expect if you're not a Freudian. Freudian theory says dreams protect sleep, but that's a counterintuitive hypothesis. Most people would think dreams disturb sleep. And so it's a nice prediction to test, because it really is, pretty unlikely that damage here, which leads to a loss of dreaming, is going to lead to worse sleep than damage here with preservation of dreaming. So that's what we predicted. There are various ways in which we measure quality of sleep, which I'll now go through with you. But first of all, let me show you the raw data in some cases. This is a hypnogram again. This is a real one, not a cartoon. This is 8 hours of sleep. And here we have the brain arousal. I have to put my glasses on so I can see it. So here you're awake, and here you're in deep sleep. Okay, and that's REM sleep, and that's REM sleep. So here's the very first patient, a non-dreaming patient, the very first non-dreaming patient that we put into the lab for the purposes of this study. The lights go out here. She stays awake, stays awake, stays awake, stays awake, stays awake for 2 hours. Then she falls asleep briefly and then wakes up and finally falls into proper sleep. But from that, she wakes up, falls asleep, wakes up, falls asleep, wakes up, falls asleep, more or less for the rest of the night, apart from this period of sleep here. And when she enters REM, she wakes up. When she enters REM, she wakes up. This is called sleep onset insomnia. In other words, can't fall asleep. This is called sleep maintenance insomnia. In other words, can't stay asleep. Here's the second non-dreaming patient. This patient fell asleep reasonably quickly. But look at this. Could not stay asleep. Woke up. Finally, a bit of proper sleep. Woke up. 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 Stayed awake. And then, the same. You see the pattern. And also wakes up from REM. Wakes up from REM. Wakes up from REM. That was our second one. Here comes the third one. Wait for it. This is the worst hypnogram I've ever seen in my life. So many people say, I didn't sleep a wink last night. But if you look at their sleep EEG, you see they slept a hell of a lot more than they think. This patient almost didn't sleep a wink that night. I mean, this is for more than 4 hours, no sleep, none. Then about 2 minutes of sleep from which she woke up, about 5 minutes of sleep from which she woke up, dipped into sleep, woke up, stayed awake, dipped into sleep. Look at that. This is absolutely terrible. And here's a fourth one. Again, New York City skyline, you see it. Lots of awakenings and waking up from REM, waking up from REM, waking up from REM, waking up from REM. So that's just to give you a picture of the raw data. These patients, the non-dreaming patients, have terrible sleep. Now I'll show you how we compared them with the dreamers. Remember, these are also stroke victims with strokes in the same area of the brain due to the same kind of pathological process. The only difference between them is these ones dream and these ones don't. It's not surprising that the dreamers, who are very ill people, these are stroke victims, recent stroke victims, their total sleep time is 375 minutes on average versus the non-dreamers, where it's 267 minutes on average. In other words, the dreamers stay asleep 75% of the night, and the non-dreamers stay asleep 53% of the night. This is a significant difference. Now, this is sleep efficiency, which is just the standard way in which we measure the quality of sleep. Again, a 20-point difference between the dreamers and the non-dreamers. The really interesting predictions are the ones that flow from that I show you now. An awakening index is how many times the patient wakes up per hour of sleep. You have to do it per hour of sleep because they have different amounts of total sleep. Non-dreaming patients wake up. These are sick patients, remember. They wake up 3.5 times per hour. The non-dreamers wake up 12.5 times per hour. It's a massively significant difference. Arousal index, which is brief awakenings. So there's standard definitions for how long the awakening is to be called an awakening versus an arousal. The dreamers wake up, they have these brief awakenings 19 times per hour of sleep. The non-dreamers 48 times per hour of sleep. Again, a massively significant difference. And the last one I'm going to show you is microarousals. These are awakenings that last less than a minute. Physiologically, on the EEG, the patient is awake, but they don't have any subjective experience of waking up and going back to sleep again. The dreamers, 15.5 microarousals per hour of sleep versus the non-dreamers, 39 microarousals per hour of sleep. So every prediction that we made as to what you would expect to find if the Freudian theory that dreams protect sleep if that theory is valid, every prediction arising from that theory that we made, that there would be less total sleep time in the non-dreamers, that there would be lower sleep efficiency in the non-dreamers, that there would be more awakenings per hour of sleep in the dreamers, that there would be more arousals, and that there would be more micro-arousals. All of those is massively strongly confirmed, at least in our pilot study, which just compares 5 patients who don't dream to 10 patients who do dream. But the difference is so vast that I think it is worth reporting it to you for the purposes of a talk like this. That's what we've found. So I've come to the end of my talk. Just to make sure you get the main message, there's 2 last things I want to say. I gave this talk recently, just before the corona thing hit, to a public audience like I'm doing now, once more, thank God, so much later. And in that audience, a colleague of mine had a little 5-year-old daughter. She hadn't even started school yet. And she listened to my lecture. And at the end of the lecture, her mother said to her, anxiously thinking she'd bored her poor little girl to tears, she said, "Did you understand what the professor was saying?" She said, "Yes, of course I do." So she said, what did he say? And so she drew this picture for her mother. On the 11th of June, 2020, she wrote, "Why do we dream? So we can have a good night's sleep." So I hope you understood what she understood, that's why we dream. It's the first time that we have empirically demonstrated a biological function for dreams. Whatever else dreams are doing, they protect sleep. Whatever else they do, they protect sleep. But the most important take-home message is that we should not exclude subjective phenomena from science. We should not exclude subjective methods of investigation from science. We will make big mistakes if we leave out actually the most fundamental property of the mind, which is that it feels like something to be a mind. We don't have to limit ourselves to the study of subjective experience. In this day and age, we can supplement it with objective methods of study, but we cannot replace it with objective methods of study. To exclude subjective phenomena from psychology, from the science of psychology, is to exclude the psyche from psychology. And that would be a terrible tragedy. Thank you very much.
So thank you for this inspiring talk, Professor Solms. We now still have around 10 minutes for discussion and questions. And we will take questions alternating from the room here and from Zoom. And if you have questions via Zoom, please post them into the chat and we will read them out loud. And if you have questions here in the room, you can just raise your hand. And we will start with a question or a comment here in the room, if you have one.
So, from my understanding, what caused this whole mess was that the REM phase was exactly the same time that we also REM, right?
Sorry, what caused this whole mess was that REM phase is exactly the same as?
Happened exactly at the same time as we are also dreaming.
Correct.
And I think you probably mentioned this, but what I haven't understood is why does this happen exactly at the same time? What controls in what phase we are currently?
Okay, so I think this is probably the single most necessary question that arises from what I've described. So I'm very glad to have that question coming straight after my talk. So the question really boils down to why is there this very strong correlation between dreaming and REM sleep if we don't know what the nature of that, what explains that correlation? Because clearly it's not the same mechanism. They're two separate mechanisms. Why do they co-activate? Why does dreaming and REM sleep happen at the same time? Why does this cholinergic surge, which arouses the forebrain, happen at the same time as this dopaminergic surge, which generates the dream process, the psychotic hallucinatory state of the dream? Well, I'll answer it first of all by drawing your attention to some other facts, which I didn't report in my talk. I told you that if you wake people up from REM, you have a 90% of getting a dream report. And if you wake people up from non-REM sleep, you have a 10% chance. That slightly spins the facts because, in fact, we have about 4 times more non-REM sleep than we have REM sleep. So although it's true that if you wake people up in non-REM sleep, you're going to only get 10% of the time a dream report. If you look at the total number of dreams that we have over the night, about 20% of them or 25% of them happen during non-REM sleep. So that's interesting, first of all. But you could say, well, it's dreams and we're asleep and they're difficult to remember and maybe they misremembered from actually having happened in the REM phase and so on. So for that reason, I want to draw your attention to this. Although it's true that on average, awakenings from non-REM sleep produce dream reports only 10% of the time. That's on average over the whole night. However, if you look at sleep onset, which is called descending stage 2, as you're falling asleep, 90 minutes before you get to your first REM period, if you wake people up then during that descending sleep onset, you have a 50% chance of getting a dream report from non-REM sleep. That can't be misremembered REM dream because REM hasn't happened yet. It's going to happen 90 minutes later. And 50%, there's a 50-50 chance that you're going to get a dream report. And then there's another phenomenon, it's called the late morning effect, which is if you wake people up in the late hours of sleep, sort of 6, 7, 8 hours of sleep, as you're heading toward a natural awakening, which is cranked up by stress hormones, cortisol increasing to get you ready for the day, perhaps to get you ready for your studies. That as you wake people up during non-REM sleep in the late morning, so you have an exponentially increasing chance of getting a dream report. And those dreams are entirely the same as REM dreams. In other words, blind raters can't differentiate them. So dreams don't only happen during REM sleep. Dreams happen during REM sleep and at sleep onset and as you're heading toward waking up. What do those three things have in common? Arousal. The brain is relatively aroused. The brain is relatively aroused as you're falling asleep. The brain is relatively aroused as you're waking up. And the brain is relatively aroused during REM sleep. So I think the most parsimonious explanation is that dreams protect sleep. When your sleep is disturbed, you dream. When your sleep is not disturbed, because that's what REM sleep is. It's a massive disturbance of sleep. That's why I pointed out to you that in my non-dreaming patients, whenever they go into REM, they wake up. So, but I don't want you to come to the conclusion from what I've just said that dreams are merely a state of arousal of the brain. Because remember, those patients who have intact REM sleep, if they have damage to the dopamine pathways, they don't dream. So it's arousal which then attracts motivational interest, then gets the dream process going. That motivational interest, that motivational response has to be triggered in order for a dream to occur. So arousal is a precondition to trigger the process of dreams. Dreams then flow from the arousal process. And I think that given everything else I've shown you, I think it really is reasonable, the most parsimonious explanation is to say that the dreams protect sleep. The best way, I'm saying too much because we don't have much time, but there you have it. That's my answer.
Okay, the next question from the Zoom audience, Lawrence Lomas, asks, does from this protection hypothesis follow that if I, as a regular person, generally dream very often, my sleep quality is good, or just that I'm probably not going to wake up very often?
Thank you very much. Since that question comes from the Zoom audience, I want to apologize to the Zoom audience for the technical difficulties you had tonight. So no, in fact, the opposite applies. That people who remember more dreams, and this has also been empirically studied, the more dreams people remember, the worse their quality of sleep is, and vice versa. The less dreams you remember, the better the quality of your sleep is. This has been studied in various laboratories, and we all come to the same conclusion, and it's been studied by various different methods. So, this finding was first published in the 1970s by John Antrobus at NYU, that the single best predictor of how well you remember your dreams is how much arousal is going on during sleep. So if dreams protect sleep, then the more dreams you're having, the more work they have to do. There's something that they're having to offset. So there's more disturbance of sleep in people who have high dream recall rate in the normal population. So I'm very sorry to hear you remember so many dreams, unless you were speaking hypothetically. It means that you're not getting great sleep. Most likely.
So since one aspect of dreaming often is that age and memory consolidation, I was wondering whether patients who are not dreaming study memory consolidation was hindered, even though their regions were not associated.
Thank you very much. So I'm glad you're asking that, because I tried to emphasize while I came to my conclusions at the end of my talk. I said, whatever else dreams are doing, one biological function that they appear to serve is that they have a sleep protection function. But that by no means implies that that's their only function. There are many things in the human body and in the human brain that do more than just one thing. And that there's often a common denominator underpinning all of those things. And if a thing is selected in for one reason and it performs some other a good adaptive function, all the more reason why it's going to be conserved evolutionarily. So there's the fact that dreaming, or the possibility that dreaming serves other biological functions, I think is perfectly likely. So the other function you're asking about is memory consolidation. And that's because there's good evidence, not quite what you said, but let me just clarify this. There's good evidence that sleep is important for memory consolidation, and that the different phases of sleep are important for different aspects of memory consolidation. But we haven't actually studied, nobody has actually experimentally studied what dreaming does for memory consolidation. And this is part of the legacy we've inherited from the erroneous conflation of dreaming with certain stages of sleep. We've assumed that that stage of sleep is dreaming, but it merely correlates with dreaming. So we need to study the effects of dreaming itself on memory consolidation. And exactly the method that I have used to study the hypothesis that dreams protect sleep, the very same method can be used to study what exactly dreams do to memory consolidation. It's going to be difficult because, as I told you, memory consolidation, the whole of sleep is important for memory consolidation in different ways. And these non-dreaming patients have very disturbed sleep. So there are confounds there. But nevertheless, there are specific hypotheses about what REM sleep has importance for non-declarative emotional memory consolidation. So to study that, I think, is something that we do need to do. And it's something that we, in fact, combining with the study that we're doing at Charité, where we're looking at the bigger version of this sleep protection study, we're simultaneously including emotional memory measures, giving the patients emotional memory stimuli and the controls, and seeing in the morning how well or badly they consolidate them. On my anecdotal observations, which is all I have so far, is that the patients who do not dream compared to the patients who do dream, stroke patients with lesions in the same area, there's no obvious difference in their memory functions, although we haven't specifically probed non-declarative emotional memory consolidation, because it requires an experiment where you give the stimulus before sleep and then you measure the recall after sleep. I'm not saying that the evidence suggests that isn't happening. I'm just saying the evidence we've got so far only shows that there's no obvious difference in the memory functions as we studied them. The reasons we studied them so far is we wanted to be sure that the patients who say they're not dreaming, that it's not actually a problem with their memory function. So we studied their memory functions very extensively, but not non-declarative emotional memory consolidation overnight. But in terms of episodic recall, which is the important thing for whether they're misremembering or forgetting dreams as opposed to not having them, there's no difference between the two groups. Thanks very much.
One last question? Or?
Okay, sure.
Okay, one last question from the Zoom audience. I'm trying to aggregate several questions at once. People are asking about the precise nature of this causation of dreams and good sleep. So what exactly is it biologically that makes our sleep better if we dream? Or couldn't it be the other way around, that it isn't that dreams make our sleep better, but that good sleep enables dreams?
Yes, and no, it's not that good sleep enables dreams, but nor is it that dreams make sleep better. Let me just be clear. The dreams are a response to something that's disturbing sleep. So your ability to dream enables you to offset disturbances to sleep. The most regular biological disturbance to sleep is REM. It is a cyclical, every 90 minutes, there's an arousal process during sleep, which is exactly why it was called paradoxical sleep in the first place. How come the brain is booting up, while you're asleep? So dreams, it's not that the dreaming makes you sleep well, it's that the dreaming prevents you from sleeping badly because there's something that's disturbing sleep. So that's the important thing to understand about what the dreaming is doing. But now we have to ask again the question, which links to the very first question that was asked this evening. But why do we have this REM surge? We know that it's not the same thing as the dream. It happens at the same time as the dream. But why is there this booting up of the brain during sleep? And it probably links to the kinds of functions that your question was about, the memory consolidation processes. Memory consolidation happens during sleep because you're not having to deal with the world. You're now offline. You can do your housekeeping. You can do your spring cleaning of your memory systems, which basically involves which memories am I going to keep, which ones need to be which ones need to be consolidated, and which ones can be forgotten. It's a sorting the wheat from the chaff. And this is an active mental process. It's not just a passive business. So there's a lot of work going on during REM sleep. So there's the fact that the brain is booting up in order to do all of these things, I think, does pose a dilemma. There's a cost and a benefit. On the one hand, we're performing an important biological function of memory consolidation. On the other hand, we're performing another important biological function, which is sleeping. Sleeping is really important. If you don't sleep, it's very bad for your health, for your physical health in all sorts of ways. And so it seems that dreaming has something to do with this kind of contradictory demands upon the organism. Simultaneously, we need to rest, and we need to do this very important mental work. And dreaming seems to be somehow enabling us to have both, to have our cake and to eat it too. Thanks very much. Thanks for inviting me, and good luck with your campaign. I really very much hope that the university maintains its long and proud tradition of, amongst all the other methods and theories that you study, you include also the psychoanalytic method and theory. Thank you very much indeed for having me.